So I predominantly work on ancient DNA, and I'm just going to introduce myself and what I've done.
You have to point it here somewhere. Yeah.
Okay. So I did a background in archaeology, anthropology, and forensics. That was me after 5 weeks of digging a hole, and I look disgusting, but it's fine. And then went to the London School of Hygiene and Medicine and studied infectious diseases. And then spent 5 years, which is a long time, doing my PhD at the Crick in ancient pathogens. And since then, I've been at UCL working with Lucy Van Dorp, who was here as well. She did a little bit of an embo stretch here as well, working again still on ancient pathogens. And the kind of focus of this work is to look at the genetic drivers of the emergence of zoonotic pathogens. And I'm going to talk about 2 pieces of work, one that I'm still doing with my PhD project, and the second one is one I'm doing here at UCL. And the first one is on Borrelia. Is on Borrelia recurrentis, which is a louse-borne relapsing fever. And Borrelia is super interesting. It's related to Lyme disease. It's a super interesting pathogen because most of the relapsing fevers are spread through ticks, and specifically looking at this one species, which is spread through lice. Yeah, and then we also don't know a lot about this pathogen in general. The louse-borne version is really hard to culture in vitro, and needs like primates to basically be able to do a lot of experiments. And there's not a lot of genetic diversity, and there's only 7 modern genomes, and they're all from one. Well, 6 of them are from one study in East Africa. Right.
Are you okay, Ben? You just laughing or joking?
Sorry, I took my headset off and then it turned on the audio. It was not related.
It's yeah, so it's tick-borne, and it's super interesting because the closest related species is Borrelia detonii, which is also tick-borne and found in East Africa, but mostly in pigs and humans. There's been a few observations in humans as well. But Borrelia recurrentis is the only known pathogen that's spread from humans to humans by lice. There are 3 pathogens in total that have done this, like switch from ticks to lice.
I don't know what's happening.
And that is, so Borrelia recurrentis is this tick-borne to louse-borne, and then
you want to use this one instead?
And then there's Bartonella quintana, which is also done this tick to louse transmission, and then Rickettsia, that one, which has also done the exact same thing. It's super interesting because they've all got this similar genome reduction. So all their tick-borne relatives are a lot larger, and they have this accumulation of these pseudogenes, which makes sense because it's massive reduction. So it's harder, it's struggling to recombine to remove all these acquired pseudogenes. And we also see an increased mortality in the louse-borne versions of these diseases. And also the host diversity is a little bit more limited with those, with their louse-borne versions of these pathogens. So in general, this tick to louse transmission is super interesting. So I'm not sure what's driving it, but it feels, yeah, it's cool. So at the Crick, we sampled and screened about 3,000 humans, and they were sequenced to about 5 to 10 million reads. And I just did some metagenomic screening, and I found 4 individuals over the last 2,000 years. So one was medieval individuals, like 700 years old, and 2 of them were around 2,000 years old, so from the Iron Age. And then there's one Canterbury or Whitefriars, sorry, they changed names, the yellow one, which is, it hasn't been dated, and super low coverage. It's only like a 0.x genome. So these, but the other ones are quite high coverage, so we can do a little bit more analysis with this. And so yeah, we authenticated edit distance damage, the standard ancient DNA, and then also when you screen for this specific pathogen and Kraken, you don't really get a lot of spurious alignments. It is very much, if you have Borrelia recurrentis, you tend to have Borrelia recurrentis. But there are closely related species, the tick-borne ones, and the chromosomes themselves are quite conserved. It's mostly on the plasmid where we see a lot of these changes between these different tick-borne species. And so we just did a comparison of the edit distance between Borrelia recurrentis and its closely related individuals, so Dettonii and Propidere, just to see, is it definitely recurrentis? And all of them are like, yes, this is definitely our individual. So these are the 4 individuals from this paper. But previous to this, in 2018, another individual was found in Norway, which was so far the only ancient Borrelia recurrentis individual found. But so there's limited, obviously, ancient data, but there's also super limited modern data, which makes it really hard to really gain an understanding of the diversity of this species. Especially if we're trying to align to our reference genomes. So this is when I came to UCL, and then we started learning this pangenomic. I don't know. Then we started learning this pangenomic approach. So we're utilizing the diversity across the whole of the read-acting fever genus species to try and understand a bit more about the gains and losses throughout this this species. And so altogether, if we just zoom into the clade that's super related, so the recurrentis and the Dettonii and the Propidere, there's about 3,000 genes across the pangenome. About like 900 of them are in the core gene. So that's that big black strip in the middle. That's like the core gene of this species. And so using this core genome approach, we can align to this reference genome rather than our single reference genome, which we tend to do in ancient DNA, because then we can at least capture a little bit more of the diversity to understand like the different historical diversity into context as well. So then just did a simple maximum likelihood phylogeny on our genomes. It's really heavily filtered for like a minimum coverage of 3x, and just polymorphic sites. And we removed a lot of the missingness of the individuals. And we found that the INH genomes tend to cluster together, and we managed to place the low coverage genome that we said was 0.x coverage with the INH individuals as well. And then we saw that the INH genome, so that's the medieval genome from Britain and the medieval genome from Oslo, also clustered together and formed a clade. So the medieval clade, and then we have our INH lineage. We're still working on this, it's still going through revisions, and then we just got the radiocarbon dates back from our low coverage individual, and it turns out it's actually medieval, even though it clusters with our genomes all the way from the INH. So I learned a very valuable lesson about not naming clades or anything based on time periods. So this is in the preprint, we put INH medieval, so I have to change this to fine. Cool. What does that say?
Stating the
Okay,
divergence.
that's what we did next. So now we also tested for homology and homologous recombination and temporality. And because we have this acquired pseudogenes, we don't see a lot of this recombination. It was like 0.1% was homologous recombination. It was super non-recombining. And it allowed us to do our temporal estimates for our divergence of the tick-borne species, Borrelia Dettonii, and we got a date of around, say, there's like maximum 5,000 years. But we also added the fishmonger's individual, and it moves the date back even further to like 8,000 years. So we can be confident, at least just with these genomes included, that the divergence date from the tick-borne ancestor is about within the last 8,000 years. And really cool is that in just before Christmas last year, Martin Sikora released a preprint of all these Borrelia recurrentis observations throughout all the data that they have. And the oldest observation fits very nicely with our divergence date, but I don't know what the sampling bias is of their data. I don't know if they have stuff that's older than 5,000 and they didn't detect it, or this is just what they kind of found. But what's super interesting is that all his observations are all Borrelia recurrentis, and none of them are Borrelia cutanei in all of his data set. And not just his data set, but our data set and any other data that I've looked at, it's always been Borrelia recurrentis. He has managed to find Borrelia crocodile, which is even like more basal, a different species, but the cutanei itself has not come up in the ancient data set. So it's something to do with this individual. Maybe it's not coming up because the reference of today's modern genome is just so drifted that it's not aligned with anything. So now we have our, how am I doing for time? Is it going over?
Very good.
Now we have our temporal timeline. We can start to use the pangenome to look at the presence and absence of genes and see if we can date them. So just a simple, just looking at the presence across this pangenome diversity that I showed earlier. And we start to see that a lot of the genes that are gained and lost tend to be involved in plasmid partitioning or recombination. And when we're dating this, a lot of, just can't go, sorry, it's been a long time since I've done this and my brain's not functioning to like the capacity. And I've inhaled a lot of xylene over the last few months, weeks. But a lot of the genes that we tend to lose over this time is involved in the recombining, which kind of makes sense because again, we know that it's lost, like it's ability to recombine efficiently and accumulate pseudogenes. What's super interesting as well is that we see that even with our ancient individuals around 2,000 years ago, we always see this reduction of 20% of the gene by our genome. So I don't know if this 20% reduction is because the cutanei has acquired a lot of genes, or whether the recurrentis clade itself is actually losing and clearing out all these genes. So this is also something that we're doing right now. It is leaning towards the reduction being due to the clearing out of the genes rather than the acquisition in the cutanei. But again, there's not enough reference, there's not enough genome and not enough data in the models to actually see the diversity of that species as well. So to summarize for the ancient Borrelia stuff, we have this like 8,000 year divergent state from this tick-borne ancestor. We can't say if any of our ancient individuals are actually tick-borne or louse-borne because of the in vitro issues. We can't actually see what genes are responsible for the switch. And it's interesting because of this, the 6 individuals in that one study, the modern genomes, they all diverged over the last like 40 to 70 years. So that's how little diversity there is in the modern data itself. And yeah, we just need more data. There's a preprint out, but still working on the actual paper. So that's that project. And now for why I'm here. Okay, so I'm currently on Lucy's I'm currently on Lucy's UKRI project, which is focused on the evolutionary drivers of malarial parasites. And it's super interesting because we're using a wide range of data. So we're using skeletal samples, we're also using DNA specimens, a little bit of everything. But we're a lot more focused on looking at the last 100 years, because this is when we see the malaria campaign, this WHO, which is like supposed to be the world's worst ever campaign to prevent the disease, because they like ran out of money. They basically started with the DDT and they started in the northern hemisphere. They worked their way down and they got to around Africa and they're like, oh, we ran out of money, sorry. And DDT is also really bad for the environment, so we're going to stop spraying all the mosquitoes. And we also see the rise of the anti-malarials as well. So we're starting to see a lot more selection, not only in the host, the mosquito vector, but also in the malaria genomes as well. And so we're looking at this and we're focusing also on the primate side. So malaria is a huge, it's such a diverse pathogen. So we've got birds, we've got primates, we're looking at everything has malaria. But our focus is on the primate data, and we're specifically looking at Southeast Asia because we see null-zi coming from the primates into the local populations. And yeah, there's like a big diversity, and it helps us answer the questions over the origins of pathogens like vivax, which is closely related to like cynomolgy, which is also a primate pathogen, and just to help us understand a little bit more about the data. And so back in I think it was like 18, 19, Lucy and the group here worked on these slides, the EBRO slides, and they managed to get some of the data, which helped us understand the spread of Plasmodium vivax into the Americas. So the project is this was the EBRO slides that were done here. And so the focus on the project is looking at these slides to try and get as much data as possible from them. So again, this is what me and Sarah have been doing in the lab over the last 6 weeks. And yeah, and they're really cool because they're old, but you can actually see the actual malaria pathogens, and I just think that's cute. But you can see from the slides themselves, there's absolutely nothing on them. But yeah, when you look under the microscope, you can actually see the photos there, and it's very cool when you work on ancient pathogens to actually be able to see the actual pathogen. I feel like that's such a unique aspect of this project. So we have probably in my suitcase, I brought over like 200, 300 slides. And I left all the anthrax at home, so you're welcome. And we are looking specifically at the primate slides, and also those that have been dated and have a good geographic location. Not all of them do, but we're going to try and use the host genome to try and work out where the primates were from. And they're all variable preservation. So some of them have these cover slips like glued onto them, and there's no getting them off. Some of them have been varnish painted, and we're trying to dip them in xylene, leave them in xylene over weeks, everything, just literally inhaling all the xylene. And we're gonna try and remove as much of this as possible. But again, when that's just the cover slip issue, we've also not 100% sure how the staining of the slides impacted the DNA preservation. So we did some benchmarking, because we don't really want to try with the samples, because some of them are absolutely beautiful and I really don't want to ruin them. This one's a Victorian slide, and even like the paper and all of this stuff, like you just don't want to touch it. And this is just one of them. There's so many different ones, and they're like sealed with tar and all sorts of weird things. But I left that one, I didn't touch it. But so the whole idea before even starting with the slides was to do some benchmarking. So we've been making up some monocultures of Plasmodium falciparum and nullii, staining them different ways and trying to re-extract the DNA, and it was mostly successful. And we've also been playing around with the protocol and designing all sorts of things. And yeah, my waist is really pretty, so I took a picture of it. Yeah, just I've never seen extract site. Yeah, it's just beautiful. Yeah, and that's pretty much what I've been doing here. And that's me, Sarah.